Hi everyone, welcome back to my class. This is Rumana Ali. I hope you all are doing well. In the last session, I have explained you about the structure of our eye, the internal structure of the eye we have seen in the last session. Let us recall all the parts that are found in the internal structure of our eye so that we can proceed towards the mechanism of vision, right? So, in the last session, I have already explained you that in the structure of our eye, three main layers are found. One is sclera, second one is choroid layer, and the third one is retina. Sclera is the outermost layer. Below the sclera, choroid layer is present, and this choroid layer, it will extend forward and then it forms some muscles, and we call that muscles as ciliary muscles. Ciliary muscles shows some thin thread-like structures that attaches to the lens of our eye. That thread-like structures are called as suspensory ligaments, which attaches to the lens. This is about the choroid layer. The third one is retina. It is the innermost and very important layer of our eye. This retina shows you the three things in it. One is photoreceptors, second one is bipolar neurons, and ganglionic cells. So, photoreceptors, bipolar neurons and ganglionic cells. These three things you can find in the layer retina. Firstly, the ganglionic cells are arranged, then below the ganglionic cells, bipolar neurons are present, and then the photoreceptors are found at the last. Photoreceptors shows you the presence of two main cells in its structure, and they are rod cells and cones. So, rods, when you see the structure of rods, they are rod-like. Two rods attaches to each other and they forms the structure of a rod. When you see the structure of cones, then one rod and one cone shaped cell, they are attached to each other in order to form the cone cell. So, rods and cones, these are found as a photoreceptors in the layer retina. Before proceeding towards the cells like rods and cones, let us first see the different parts that you can see in our eye. So, besides these three layers, you can also see the presence of a part that is present in front of the lens. The lens is behind a part which is called as iris, and iris is a colored ball that you can see. So, the iris, it shows one hole in its structure. In the center you can see one hole through which the light enters into the eye, and that hole is called as pupil, and over the iris and pupil a transparent covering is done. A transparent covering can be seen, and that covering is termed as cornea. So, this is about the different structures that you can find in the human eye. In the layer retina, besides photoreceptor, bipolar neuron and ganglionic cell, you can also find two main spots. One spot is the blind spot where the light doesn't fall, and if the light falls also, this spot will not give you the vision. This is called as blind spot. And one other spot is present which is called as yellow spot, where if the light falls, then it will give you the bright vision. We'll see why the yellow spot provides the bright vision, why the resolution is high in the yellow spot. That we'll see within a minute. And this yellow spot can also be termed as macula lutea. This yellow spot shows you the depression, and that depression can be termed as fovea. These are the different parts that you need to remember. Now, as I told you that bipolar neurons and ganglionic cells are present in the layer retina, these bipolar neurons and ganglionic cells, they take the message and they will move to the peripheral nervous system. So, the way for the nerve cells is the blind spot. The nerve cells can come out of the retina. They can transmit the message by the way called blind spot. So, blind spot is a way for both nerve cells as well as for the blood vessels. The blood vessels will enter into the eye through the blind spot, and the nerve cells will move out of the eye through the blind spot only. And these nerve cells, when they combine together, they form a nerve. We call that nerve as optic nerve. Now, we'll talk about the photoreceptors. I told you that photoreceptors contains two cells, rods and cones. Let us see what are the different pigments that you can find in these kinds of cells. When you look at the cones, then a protein named rhodopsin is found in the rods. And cones shows you the presence of so many pigments in it like chloropsin, cyanopsin and erythropsin. These different pigments helps us to visualize the different kinds of light. Now let us move to the differences of rods and cones. Rods are the cells that becomes activated in the dim light. During the night time, the rods, they get activated and they help you to visualize the different things that are present in your environment. So this type of vision is called as photopic or twilight vision, and it does not show any color. No colored vision is provided by our rods. The next one is cones. Cones are the cells that gets activated in the bright light. During the daytime, these cones, they becomes active, and due to the presence of the different pigments in its cells, they can be able to provide photopic vision. Photopic vision means we can be able to visualize the different things present in our surrounding. Their color we can visualize due to the cells called cones. So it provides the colored vision to the individual. These are some of the differences that you should understand about the different photoreceptors called rods and cones. Now, I told you that yellow spot, it is a spot where the resolution of the light is very high. You can be able to visualize the different things in a bright manner, in a very clear manner when the light falls on our fovea. So why it happens? What is the reason? So when you see the fovea, it's a depression. I told you that it's a depression in the retina layer, right? So this depression contains more number of cones, and cones are the photoreceptors that is responsible for photopic vision. It shows you the colored objects. It provides the colored vision. So due to the presence of more number of cones in the fovea, the resolution will be high, and you can see the picture or anything in the surrounding in a very clear manner when the light falls on the fovea. But when the light falls on the blind spot, it will not help you to visualize anything, because blind spot is a spot which is away for optic nerves and blood vessels. It does not provide any vision to our eye. This is about the different parts, different layers, different photoreceptors that are found in the human eye. Let us talk about the mechanism of vision. We have already seen that in the layer retina, ganglionic cells are present. Ganglionic cells are present, bipolar neurons are present, and even rods and cones are found. In the rods, you can find the presence of a pigment called rhodopsin. In cones, the three different pigments are present: cyanopsin, erythropsin and chloropsin. Okay, these are the different pigments. These all pigments like rhodopsin, cyanopsin, erythropsin and chloropsin, they contain one substance and one protein in its structure. The substance which they contain is called as retinal, and the protein which is present in all of these pigments is termed as opsin. So in the pigment you can find the presence of a substance retinal and a protein called opsin. In all the pigments, these two things can be found. Now, we know that light always falls on the layer retina. So this is the light. It enters through the pupil and it directly falls on the retina. In the retina, ganglionic cells, bipolar neurons and photoreceptors are present. But when the light falls, ganglionic cells and bipolar neurons, they do not do anything. The light directly enters into the the light directly receives by the photoreceptors. When the photoreceptors receives the light, then the photoreceptors are rods and cones. In that rods and cones, pigments are present, right? So that pigments, due to the absorption of the light, that pigments will split. So the pigment, it contains opsin as well as retinal in its structure. When the pigments receives the light at that time, the bleaching of the protein and retinal takes place, the separation of the substance and the protein. It can be seen, which is called as bleaching. Now, due to the bleaching of the pigment of the photoreceptors, the action potential can be seen in the bipolar neurons, and in the ganglionic cells. Due to this action potential, they becomes activated and they start to transmit the signals to the brain. So, after the bleaching of the pigment of photoreceptors, the action potential can be seen in the different neurons. Due to the action potential, the different neurons, they start to move out through the blind spot. They will move out, they form an optic nerve, and this optic nerve carry the signals towards the brain. It carries the signals to the brain, and then the brain commands the eye. It transmits some other neurons. It commands the eye to see, to visualize the thing which is present in the environment. This is how the vision can take place due to the bleaching of the pigment of the different photoreceptors, and due to the action potential of the neurons. The neurons takes the message to the brain, and then brain sends the command to our eye in order to visualize the different things that are found in our surrounding. So, this is about the mechanism of vision. Let us move to the next topic, and our next topic is adjustments in iris. We know that our ball that resembles from outside, the colored ball, that is called as iris. So, what happens due to the visualization of the different objects to the iris? Let us see that. So, the iris, it shows you the two main muscles in its structure. Firstly, the iris is present, and between the iris you can find a hole that is called as pupil, through which the light enters into our eye. Now, this iris shows you the two main types of smooth muscles. One is circular smooth muscles and the other one is radial smooth muscles. Circular smooth muscles are circular in shape, and radial smooth muscles are like rods. So, these are two different kinds of muscles that you can find in the structure of iris. Due to the movement, due to the contraction of these muscles, you can be able to see the different objects in the light and also in the presence of dim light. So, whenever the contraction of circular smooth muscles takes place, when the circular smooth muscles gets contracted, they gets contracted when you see the object in the presence of light. So, when you are present, when you entered in a lighted room and when you are trying to see the different objects that are present in your environment, at that time the circular smooth muscles of the iris, they get contracted. Due to the contraction of the circular smooth muscles, the pupil which is present, it gets smaller in size. So, the pupil, it gets smaller to visualize the different things in the presence of bright light. But when you move into the dark room and when you are trying to see the objects that are present in the dark room, then the radial smooth muscles which are present in the iris, they get contracted. Due to the contraction of the radial muscles, the pupil which is present, it becomes larger, it gets dilated. So, to dilate the pupil, the radial muscles, it gets contracted, and due to the contraction of the radial muscles only, the pupil, it get dilated, it becomes larger in size, due to which we can be able to see the different objects that are present in the dark room. Not clearly, but we can see the objects that are found in the dark room. This is how the contraction of circular smooth muscles and the contraction of radial smooth muscles regulates the size of the pupil and helps us to see the different things in the presence of bright light and also in the dim light. This is about the adjustment in iris, and this adjustment in iris is not under our conscious control. It is an involuntary function. So it happens due to the contraction of circular and radial smooth muscles. Let us move to the other topic, that is adjustment in lens. We know that behind the iris, lens is present. Here you can see this is our lens. Lens gets attached with some fiber like material, and that thread like or fiber like material is called as suspensory ligaments, and these suspensory ligaments are attached to the ciliary muscle. So now the contraction and relaxation of the ciliary muscles regulates the position of the lens, regulates the adjustment in lens. Our lens is in convex shape, so we call our lens as convex lens. Now let us see what will happen if you want to see the object that is present far away from you. If the object is far away from you, then the lens, it is stretched and the curvature of the lens becomes less. How it happens? It is due to the relaxation of the ciliary muscles. Whenever any object is present far away from you, at that time the ciliary muscles, they get relaxed, and due to the relaxation of the ciliary muscles, whatever the suspensory ligaments that are attached to it, it stretched, and due to the stretching of the suspensory ligaments, the lens also gets stretched and the curvature of the lens becomes less. That means the lens has less curvature in the case when the object is far away from you. In the case 2, if the object is nearby, it is very nearby, and if you are trying to see that, then the lens, it increases its curvature. The curvature of the lens becomes more, and the lens has more curvature in the case 2 when the object is kept nearby to you. In this case, the ligaments which are present, the suspensory ligaments, they get relaxed due to the contraction of the ciliary muscles. When the ciliary muscles contracted, these suspensory ligaments, they move towards the lens, right? They would not stretch. As the suspensory ligaments are not stretching, they are relaxing. Due to the relaxed ligament, the lens has more curvature, and as soon as the lens shows you the more curvature, the objects which are kept nearby you, you can see their objects clearly. So this is what happens to see the objects which are kept far away and near by us. So if you want to see the object that is present far away from you, then relaxation of the ciliary muscles would happen. Due to the relaxation, stretching of the suspensory ligaments takes place, and due to the stretching, the convex lens, it also gets stretched. Now, if the object is nearby you, then the ciliary muscles, they get contracted. Due to the contraction of the ciliary muscles, they becomes loose, they would not stretch, and as the suspensory ligaments would not stretch, the lens will also not stretched, and the lens shows the more curvature. It has the more curvature in the case 2 if you want to see the object that is nearby you. So these are the two situations or cases in which you can see the different adjustments in the lens, due to which the visualization of the different things is possible. So this is about the adjustment in iris and also about the adjustment in lens. Up till we were discussing about the human ear and the mechanism of vision. Now let us move to the other sense organ of a human body, that is human ear. So human ear is divided into 3 main parts, that are external ear, middle ear and internal ear. External ear shows you the different parts. External ear shows you the different parts like pinna, auditory meatus and tympanum. Middle ear shows the presence of ear ossicles in it, and internal ear shows the presence of bony and membranous labyrinth. Okay, so these 3 parts of the ear forms the internal structure of forms the whole structure of the human ear. Let us see the diagram to understand all these 3 parts of a human ear. So the part which resembles from the outside, that is covered by the skin, we call this part as pinna. This is extended and it shows a tube-like structure. We call this tube as external auditory meatus. After the external auditory meatus, one structure is present like this, and it is termed as eardrum. Eardrum can also be named as tympanum. So in this way, three parts forms our external ear: pinna, external auditory meatus, and eardrum, that is also called as tympanum. This is our external ear. In the auditory meatus, hairs are present. Along with the hairs, some glands are found, and we call that glands as cereminous gland. Cereminous glands are responsible to secrete a wax-like substance, to secrete a wax into the external ear, and we call that wax as ceramen. So ceramen is a wax that is produced by the glands which are present in the external auditory meatus. Wax and hairs, these two things are present in the external auditory meatus that does not allow any foreign material to enter into our ear. This is how it provides protection to the middle and internal human ear. Now, when you see the middle ear, this part is called as middle ear. Then it shows the three bones, three main bones are present in the middle ear. One is malleus, which is also named as hammer. The other one is incus, which is also termed as anvil. And the third bone is stapes, which is also called as stirrup. Malleus, incus and stapes. These three bones are found in the middle ear of a human being. And stapes is the smallest bone that is found in the human body, which is also termed as stirrup. So this stirrup is found in the middle ear. When you put any drop into ear, sometimes it will come into your throat. So how it can be possible? What is the reason behind this? This is because of a tube present in the middle ear. One tube is arising from the middle ear, and this tube opens into the pharynx. And we call this tube as eustachian tube. So here, external ear shows you the pinna, external auditory meatus, and eardrum, which is also termed as tympanum. Middle ear shows the three main bones in it: malleus, incus and stapes. Stapes is the smallest bone that is found in the human body. After the middle ear, and middle ear shows the extension of one tube. One tube is arising from the middle ear, and we call that tube as eustachian tube, which opens into the pharynx. After the middle ear, internal ear is present, and this internal ear shows the two main parts, bony and membranous labyrinth. So this internal ear is an important part of the ear, because the function of hearing can be regulated by the internal ear only. So we need to understand the internal ear in a deep manner. So in this class we have learned about external ear and middle ear. In the next session I'll explain you about the internal ear. So that's all for today. Meet you in next class. Thank you.